Good morning, everybody. Welcome. We are about to begin the session on biophysics of learning and computing. I'm very pleased to introduce the first speaker, Yiota Poirazi from the Institute of Molecular Biology and Biotechnology at the Foundation for Research and Technology in Crete. Come on.
Well, thank you very much for the invitation. It's great to be here. And I want to talk to you about what my lab has been doing for a long time now, which is mainly focusing on trying to understand how dendrites contribute to different functions at the neuronal and the circuit level. So dendrites, what do we know about them? Their name comes from the Greek word dendron, which essentially means tree, and that's obvious because they look like trees. And they were first described by Camillo Golgi in 1873. At the time, he used his famous Golgi technique, and they believed that they served a nutrient role, so providing nutrients to the neurons. And it wasn't until 1888 that Ramon y Cajal proposed the neuron doctrine, according to which neurons are the processing units of the brain, and dendrites are essentially the entry to the neurons. They serve as their input space, and axons serve as the output. So in this talk, we're primarily interested in active dendrites. What are active dendrites? So active essentially means that these structures have an ability to change the way they process their incoming inputs. And incoming inputs can be processed in different ways. For example, the most known common one is a simple linear summation, which means that as the input strength or number increases, the output is just a linear function of the input, which is shown here in black. But there are other ways of influencing the way the structures compute. For example, as you can see here in green, we can have supralinear integration, which means that as you increase the strength of the input, the output deviates from the linear curve and it goes above it, so it can become supralinear. And we can have other cases where the opposite happens. So as you increase the strength of the input, the output saturates, so it's below linear. So we have all these types of configurations in dendrites. And the ability to deviate from linearity is usually attributed to the presence of voltage-gated ionic conductances that can either boost or suppress the response of the branch locally. So why do we care about dendrites? Well, first of all, because they have these nonlinearities that allow them to perform nonlinear computations and therefore increase the ways they can process signals. But also because there is a lot of information out there that their anatomy, biophysics, and plasticity properties are altered in various neurodegenerative conditions. Therefore, we believe that if we understand how dendrites contribute to these functions, maybe we'll be able to come up with new ways to treat these diseases. How do we do that? In my lab, we use the mostly computational approaches, although we collaborate a lot with experimentalists to validate our models and explain experimental data. And we have many different kinds of models that vary in their level of abstraction. We have models that simulate the way single neurons process information in the hippocampus and the cortex, prefrontal cortex and visual cortex. Recently, we also developed models of interneurons in the PFC and the hippocampus at the single cell level. We also have models that operate at the microcircuit level, so they consist of a few neurons, either detailed in their morphology or reduced in their morphology, connected into small microcircuits of a few hundreds or tens of cells. And we also have larger scale models like this one, which simulate entire areas. In this case, could be unassociated, not entire areas, a large portion of a brain area, let's say, which could be, in this case, an associative area, and we implement plasticity rules in these models so that we are able to train them to simulate learning and memory. Okay, so I've always get this question of what are these models good for, especially if they're only theoretical, what can they help us with? What are the kind of information that they can provide? So I want to tell you a little bit about what we have learned from these models in the previous years. And I want to start with my early work, which was purely theoretical, and then move on to more recent work, which also combines experiments. So this is very early on when I was a PhD student in Bartlett Mill's lab. And at the time, we wanted to figure out how dendrites integrate incoming signals. It was not known yet whether they are able of generating nonlinear outputs. So we assume that they can perform local nonlinear computations because of the presence of voltage-gated conductances, which now we know for sure. We assume that the axons of presynaptic neurons can move around, they can rewire, so as to find better suited partners. And we assume that this rewiring can be activity dependent. So where to project depends on the activity of those synapses. Based on these assumptions, we generated a single neuron model. It's a very abstract concept. It's a mathematical model where we implemented these properties and we used them to train this neuron on a classification task. So to teach it to discriminate between, say, cats and dogs, right? And what came out from these assumptions, the local plasticity rules, was a prediction that axons that fire together will end up wiring together, much like what Hebb said, but at the local level, so that they would wire together within the same dendrite. And this was because if they do that, this is a much more efficient way of activating the local nonlinearity, okay? So this was a prediction that came out in 2001. And about a decade later, we have strong experimental data supporting this prediction, which essentially led to some kind of clustering of inputs within the same branch, right? This was the outcome of this prediction. And we now know from a lot of experimental studies that this clustering does happen in the brain. It happens in vivo. It happens as a response of plasticity and learning. And it's now a fundamental principle that has at least been described. So this is an important contribution of how a model can help generate a prediction that will then steer the interest of the experimental community. So how does this clustering help the brain? In the same study, we also measured what would be the change in the ability of this network to learn many such patterns of cats and dogs. And we figured out that if you have these nonlinear dendrites and the ability to rewire, then the increase in the storage capacity of this model was about an order of magnitude higher than having linear dendrites. So a major advantage of having dendritic nonlinearities is the ability to compute more information. And more recently, we also were able to show that having nonlinear dendrites in which synapses can form clusters can also serve as a mechanism for linking memories across time. So this was a nice experimental study done in the Alcino Silva lab, where they showed that if you have two memories learned within a time period of a few hours, then the second memory benefits from the learning of the first. And this is because they both converge, they're both stored in overlapping neurons, overlapping neuronal populations, which is shown here in red. So we used our models to show that this neuronal overlap that underlies the interaction between multiple memories is a result of forming cluster synapses within the dendrites of these common neurons. So this is a number of let's say, outcomes that came out from this early work, highlighting why it is important to do this kind of modeling. Another work that I want to briefly introduce, because it is the basis of the more recent stuff that I will describe, is what I did with Bartlett again back then with respect to simulating single pyramidal neurons in the hippocampus. So as a PhD student, no, actually I was a postdoc at the time, I built a detailed C1 model of the hippocampus, a pyramidal neuron, that contained a large number of the ionic and synaptic conductances we knew that these cells express. And we used this model to figure out how the dendrites of these neurons integrate signals. At the time, it still was not clear what would be the advantage of having, let's say, complicated dendrites in these features. The prominent idea was that pyramidal neurons still compute like linear point neurons. They summate their inputs and they produce an output. So we were wondering whether this is the case. And to highlight this possibility, we tested in our neuron whether the location of the inputs matters. If this was a linear point neuron, the location would not matter, right? So we grouped synapses within dendrites or separated them between different dendrites and look at the response of the cell body. And what we found out was that if you group the same number and strength of inputs within a single dendrite, then the response is the one dictated by the red curve. So it's a sigmoidal response, and you have a boosting of inputs within this regime that then reaches a saturation level. This is not the case if you separate your inputs into two different dendrites, in which case the output of the cell is just a linear summation. So this was the first prediction that dendrites of C1 pyramidal neurons integrate signals in a nonlinear manner. And we're very happy that this prediction was verified experimentally soon afterwards, like a year later, in the lab of Jackie Chiller, which showed that in the basal dendrites of neocortical pyramidal neurons, you have exactly the same situation. Within the branch, you get a sigmoidal integration. Across branches, you get a linear summation. And a few years later, in the lab of Jeff McGee, for the same neurons that we used the model for, so the same CA1 pyramidal neurons. So this is another contribution of the modeling work. Now, why is it important to have nonlinear dendrites? Well, because then we can think of a neuron as something similar to a neuronal network and not a point neuron. So in this case, we showed that we have nonlinearities within each branch, right? And these nonlinearities look like a sigmoid. If you're familiar with artificial neural networks, this is what typically an artificial neural network looks like. So you have a transfer function, which in this case is a sigmoid, and you have multiple of these transfer functions in each one of the dendrites, and then another thresholding nonlinearity at the cell body. So you can think of describing the neuron as a two-stage neuronal network where each dendrite contributes a local nonlinearity at its own level. And based on this assumption, we showed in that paper that this reduced mathematical abstraction can very well describe the output of the detailed model in terms of its mean firing rate. So you can use a two-stage artificial neural network to predict what will be the output of this biophysically detailed neuron for many different numbers and arrangements of synaptic inputs, which is a prediction that has not been exactly validated yet, but we have a lot of supporting evidence for it with respect to what the dendrites do. And it has helped shape our understanding of how pyramidal neurons in the cortex generally operate, in the cortex and the hippocampus. We started out with a two-stage neuronal network, but others have also spoken about more complicated structures where each of the trees, like the basal and the apical tree, may be doing its own nonlinear computations. So now we believe that pyramidal neurons in the cortex and the hippocampus are much more complicated than we thought 40 years ago. Okay, so that was the old work, and I now want to move into the more recent stuff, which have to do with interneurons. So we know a lot about pyramidal neurons. This is what we described earlier on. But for interneurons, still the dominant hypothesis is that they simply summate their inputs, and if they pass a threshold, they generate an output. And there is a lot of experimental data coming out the last at least 1 decade, suggesting that the dendrites of these neurons also may have interesting properties. For example, we know that the CA1 stratum radiatum interneurons, their dendrites have the ability to generate local dendritic spikes in response to increasing number of inputs, which is shown in red here. And therefore, the output of these cells looks something very similar as the pyramidal neuron. So you can have a sigmoidal nonlinearity in the dendrites of this type of interneurons. Also, in other type of interneurons, in the cerebellum, for example, you can have dendrites that respond in a similar supralinear manner if you look at the concentration of calcium. But if you look at the EPSP, then the response is sublinear. So you have two types of nonlinearities in these branches. And one of the most dominant classes of interneurons that we know of is what is called the fast spiking basket cells. It is one of the most important interneurons in terms of the number of its population, and also because it regulates the somatic firing rate. And in these neurons, there is also evidence that the dendrites can generate supralinear responses, right, which is what is shown here, both with respect to calcium and voltage. And this is data generated by the lab of Balázs Roxa. So in light of the information that interneurons also have dendrites that can compute in nonlinear ways, one would wonder whether the point neuron dogma is still a valid one. Is it still true that an interneuron is just summing its input and producing an output according to a linear point neuron? So to answer this question, a talented master student in the lab, Alexandra Zilivagi, she wanted to follow my steps in doing a similar approach. I told her no, but she insisted. So what she did was she essentially took morphological reconstructions of these cells from the hippocampus and the prefrontal cortex, 8 morphologies in total, and she built biophysical models similar to my C1 pyramidal neuron, where she incorporated the ionic conductances and synaptic mechanisms that are present in these cells. And once she validated her models against a bunch of experimental data that I'm not showing for the interest of time, but this work is on bioRxiv, so you can look at it, she used this model to generate predictions about how the dendrites of these cells compute. And what she found in the same experiment, where you increase the stimulation of the dendrite, the number of inputs that go in, and you look at the response in the dendrite of the cell body, she found that indeed some of these dendrites are able of generating dendritic spikes, so they have a supralinear response, but others in the same morphology are not capable of generating a local dendritic spike, and therefore their response is sublinear. So the model predicts that we can have both types of nonlinearities with respect to voltage, at least, in the same morphology, and this was true for all of the 8 morphologies we tested. And she also went one step ahead to figure out what is it that underlies these nonlinearities. One should take into account that these interneurons in particular do not have strong NMDA conductances, which is the main contributor in the pyramidal cell. They do have, however, AMPA receptors that are calcium permeable, and they do have sodium channels which are much lower in the dendrites than the cell bodies. They are an order of magnitude, so 10 times less in the dendrites. So she looked into the virus biophysics, and she found that it was the sodium that enables some of these dendrites to generate spikes, because if you close it, the spikes are no longer there. What is intriguing is that we use the same distribution of ionic mechanisms in our models. So all of our dendrites have pretty much the same or similar values of sodium conductances. So why is it that some dendrites are supralinear and others are not, if the biophysics are very similar? So we looked at what could be another determinant of this function, and obviously the next thing to consider is morphology. So we looked at the morphologies of the 2 types of dendrites in both areas, and we looked at a lot of features, but the ones that came out to be significantly different, in the hippocampus, it was the diameter. So supralinear dendrites have a larger diameter. In the prefrontal cortex, it was not the diameter, but it was the length. The length was significantly different between these 2 types of dendrites. Both of these features constitute another measure, the volume, which considers both length and the diameter. So we looked at the volume, and we found out that the volume is statistically different between these 2 classes, and as a result, the input resistance to these dendrites is also different. So we have morphological determinants that tell us how these dendrites will integrate in their inputs. They're not precisely the same in both areas, but they both involve the volume. So in the hippocampus, we have thicker supralinear dendrites. In the PFC, we have longer supralinear dendrites. Now, why is it important or interesting to have 2 types of integration in the dendrites of these cells? So we know from our work in the pyramidal neurons that if you have nonlinear dendrites, then the location of inputs matters. So we went ahead and distributed synaptic inputs onto the branches of these morphologies in many different ways. So we randomly placed them on all dendrites, or we organized them within a few dendrites, and we asked, what is the response of the model in these changes in the arrangement? And we found out that these cells, both in the hippocampus and the prefrontal cortex, they prefer to have their inputs distributed in a scattered or dispersed manner. So they get a much higher response if the inputs are distributed randomly than if they're clustered within a few branches, which is counterintuitive because in pyramidal neurons it is exactly the opposite. And one would expect that if you have supralinear dendrites, then it makes sense to group your synapses in a few branches to activate these nonlinearities. But this was not the case in the interneuron. So we wonder, why are we getting this phenotype? So there are a few things that one has to consider. As I said, these branches do not have NMDA receptors. So they don't have a long-lasting depolarization that would boost their signals. They also have a very high conductance of potassium channels in their dendrites, which is a hyperpolarizing conductance that can counteract the sodium, the fast sodium spike. And then the last thing that we've seen is that morphology is important. They have very thin dendrites. So we took in all of these factors into consideration. And we saw that depending on how you organize your synapses, then different contributions come from these factors. For example, red is the morphology and orange is the A-type potassium channel. So you look that if you thicken the dendrites, if you increase the dendrites, then the dispersed arrangements have a serious drop in their ability to generate high firing rates. Whereas the cluster ones do not have such a big change. So the cluster ones do not suffer so much from the morphology, but they do suffer from the A-type channel. Because if you close the A-type channel, then the response suddenly increases massively. So this is the way to think about this. Dispersed arrangements are weak. You have small inputs. If you have a very thin dendrite, then you don't have a serious signal attenuation. It gets to the cell body, so it's sufficient. If you have clustered inputs, you strongly activate your local conductances. You generate a sodium spike, maybe, but this also strongly activates the potassium channels, and therefore you have an immediate hyperpolarizing force that shuts this response off. So there is a trade-off between both of these two mechanisms. And as a result, if you close them both, you eliminate this dispersed preference, which is what we see here. Okay? So we have a bit of a better understanding as to the mechanistic basis of this preference. Okay, so we know they have nonlinear dendrites. We know they're sensitive to the arrangement of the input. What is a good mathematical description of such a cell? Is it still that the point neuron is a good abstraction, or do we need a different one? So to answer this question, we took the morphologies and we mapped them into two theoretical abstractions. The first one is the linear point neuron, where your dendrites essentially have just a linear transfer function, and what goes in is the number of inputs in all dendrites. And we also have a second version of the model where you break these two hidden layers into supralinear and sublinear ones, each one taking input from the respective dendrites. And we compare how good these two abstractions are with respect to predicting the output of the neuron. And what we find is shown here. These are two representative examples for the hippocampus and the prefrontal cortex. And you can see that the two-stage ANN can predict the firing output of this cell in a much better way, in the sense it has a much higher correlation coefficient when you feed the data, than the linear model, which admittedly is not terrible, right? It still does a relatively good job, but it fails to capture, the end of the nonlinearity. So the two-stage ANN is better, and it also considers the the prediction that the dendrites of these neurons are nonlinear. And we've done this for all of the 8 different morphologies, and it came out true that in all cases, the two-stage nonlinearity is better. Okay? So we propose that if you want to describe these cells by considering what their dendrites do, it is better to assume that it is a two-stage neuronal network, let's say, with 2 types of nonlinearities, rather than a linear point neuron. So why is this important for the brain? Why would the brain go into all of this trouble to come up with fancy interneurons if there wasn't some kind of an important advantage? To answer this question, we used our previously developed large-scale network model of an associative area, where we have the pyramidal neurons described as simple integrate and fire nodes with just a few supralinear dendrites. And now we add into this model a reduced version of the simple interneuron, which now we extended with dendrites, a few dendrites. Some of them are supralinear, others are sublinear. And we ask, what is the advantage with respect to learning a new memory in this network? How do we learn a new memory? We integrate a number of plasticity rules in this work, and this is published, so I'm not going into detail. So essentially, you have local LTP and LTD, you have homeostatic plasticity, plasticity of neuronal excitability, and axonal rewiring. So if you have all these features in the pyramidal neurons of this network, you can train it to learn various, should be playing. Well, anyway, this was supposed to be a nice, cool video, but it's not playing. So you can train the model to form memories by strengthening and weakening the inputs. And what you see there as dots are essentially the synapses for multiple memories. Each memory is an associative memory, so it consists of 2 components. And over time, the network learns, and you end up with a subpopulation of neurons that is forming the engram. This is the neurons that encode this memory. So we can look at the properties of these engram populations with respect to having a linear interneuron, a nonlinear interneuron where the inputs are dispersed or clustered. And what we see is that the size of this engram is bigger if you have a linear interneuron, and it drops down to a much smaller, by more than 20%, if you add these dendritic nonlinearities. These engrams also fire at lower firing rates, so this is the linear, this is the best nonlinear, let's say, and they are much sparser. This is the linear, this is the nonlinear case. So lower firing rate, sparser engrams, smaller engrams. What does this mean? It means that the network is able to learn exactly the same information as before, but using fewer resources. So the suggestion that we make here is that these 2-stage nonlinearities in interneurons are providing resource savings in our network models. So in summary, at the single cell level in this work, we predict that the dendrites of fast-spiking basket cells can come in 2 flavors. They can have both supralinear and sublinear dendrites. Morphology is a key determinant for the difference between the 2 dendrites. And these nonlinearities make these neurons slightly more complicated than we thought. And we suggest that a 2-stage nonlinearity or a two-stage artificial neuronal network is a better description, a better abstraction for these interneurons than the linear point neuron. And at the network level, these nonlinearities allow associative areas to learn information by using fewer resources. Okay, so in this work, I talked to you about how biophysical models can contribute to our understanding at the single cell level, how we can take this information to compute the function of a larger network level, and now I want to tell you a little bit about the microcircuit level. So this is work that was done in collaboration with Attila Lozonsky in Columbia University, and it just came out in Neuron, and it's a combination of modeling and experiments. The goal here was to understand how VIP interneurons control the dendrites of pyramidal cells in the CA1 pyramidal, in the CA1 area, with respect to spatial learning. So the VIP interneurons are mainly disinhibitory. They control the somatostatin cells, which then project to the dendrites of the apical tree of pyramidal neurons, and they control how the apical tree communicate with the cell body. This is one class of VIP cells. There is another one that projects at the cell body. So the question that Attila and I were mostly interested in is to understand how these cells contribute to spatial learning. To do this, Attila trained mice to run down a treadmill with various features. He placed rewards at various locations, and he imaged the activity of both pyramidal neurons and VIP interneurons during learning. And he found out that these interneurons come in two flavors. So their activity is modulated with respect to the motion of the animal, and it can be modulated in two ways, in a positive or a negative manner. Positive means that while the animal runs, these interneurons fire more strongly. Negative means that when the animal stops, these neurons fire more strongly. So you have both types of these interneurons in the animal. And their relative proportions is approximately 80 to 20%, with the positive velocity ones being the dominant class. We don't know much about what is a positive velocity or negative velocity class with respect to their molecular markers, right? We just know they're velocity modulated. So the question we wanted to figure out then is how, or if any, this type of interneurons contribute to spatial learning. So in this case, the reward was randomly placed, so there was not much learning in the animal. So what Attila did was essentially to place the reward in a particular location and train the animal over a period of 3 days to learn where this reward is located on the belt. And they measured learning by looking at the fraction of the leaks that the animal was doing in that particular location. So the more the leaks, the more likely it is that the animal learned the location very well. And they imaged, as we said, the pyramidal and the VIP interneurons, and they found that these positive and negative modulated classes, they grew bigger. So there was a larger percentage of VIP neurons that now were modulated by velocity, either in a positive or in a negative manner. And at the same time, if you look at the C1 pyramidal neurons, what they see is that before learning, the animal maps the entire space very well in a uniform manner. But after learning, there is a very large number, well, 30% increase in the number of C1 pyramidal cells that fire in locations that are close to the reward zone. So there is an overrepresentation of the reward zone in the CA1 pyramidal neurons. And the question is whether there is a link between these two observations, the increase in the VIP neuron modulation and the learning of the reward location. So to do that, what Attila did is essentially to train the animal in a double task. So first, you learn the location of the reward in one task, and then they shifted the reward to make the task more challenging to a new location. And they retrained the animals to learn this new location. And during this experimental phase, they performed optogenetic manipulations, where they silenced or excited the VIP population. They could not discriminate between the positive and the negative velocity ones, because we don't have markers for those, so they excited the entire population. And what they found out was that, as just to remind you from the previous slide, during control case, you have an overrepresentation of the reward location, and if you silence the VIP interneurons, this overrepresentation disappears, which means that the VIP cells are certainly contributing to the learning of the new reward location, but it is still unclear how they do it and which of the two subclasses is contributing. So that's when we teamed up. And this is the work of Spiros Havlis, a very talented postdoc in the lab. And what Spiros did is to build a detailed, well, not detailed, it's morphologically reduced and biophysically relatively detailed model of a CA1 network, microcircuit, where he incorporated 6 types of interneurons, and importantly, the 2 types of the VIP cells that we know, the ones that express calredinin and the one that express CCK. No, what's the CCK? I forget. Another marker. So we have these 2 types of interneurons in our model, and we wire them up into a microcircuit that looks like this. So we have the pyramidal cell and the 2 types of VIP interneurons connected. One goes to the cell body, the other one goes to the somatostatin interneurons. And we use this microcircuit model to reproduce pretty much the same behavior that we've seen in experiments. So to do that, we give input to the microcircuit from the entorhinal cortex that has the shape of a grid cell. And we know that if these grid cells align, then the output of the pyramidal cell will be a place cell. So this is what comes from the entorhinal cortex. And we simulate the learning paradigm by describing a virtual mouse that goes down a virtual belt and learns the location of a virtual reward. All this is done in silico. And then we compare the results that we get from the experiments with the results that we get from the simulations, and we're calibrating our parameters so as to reproduce the experimental data. So in the control case, there's a uniform mapping of the space. Post-learning, there is an overrepresentation of the reward location in the network. And if you silence all VIP neurons, then this goes away. And the interesting question that comes up, that the model can produce is which subtype of the VIP interneurons is contribute to this modulation. So we can selectively block only the CR or only the CCK type of interneurons and look at what happens in the network model. And what we find is that if you block the disinhibitory population, which is the CR, then this phenomenon, the overlearning, the representation of the reward disappears, whereas if you block the CCK interneurons, this doesn't go away. In fact, it gets enhanced. So the main output from this work is that we can identify, let's say, the subclass of these interneurons that are causal with respect to the phenotype that we well, causal. They're responsible for preventing the learning of the reward location. We still don't know if it's really causal. And the model can also tell us why is this a case, because we can look into the fraction of place cells that we have before and after learning, and we can also look at the firing rate of these place cells before and after learning while we perform various manipulations, closing all VIP interneurons, one or the other subclass, only the positive velocity modulated or only the negative velocity modulated. And we can see from here, from these graphs, that essentially the reason why the disinhibitory neurons are the most important ones is that they control both the firing rate of pyramidal neurons and the number of place cells that are formed after learning. So the model can provide a mechanistic explanation for the phenomenon that we've seen in experiments. Essentially can explain why the disinhibitory class is the one controlling the learning of the reward location by incorporating these various versions of interneuron subtypes. So in conclusion, what we've shown in this work together with Attila is that VIP interneurons come in different flavors. They can be modulated by the locomotion of the animals in both positive and negative ways, and that the disinhibitory action of these VIP interneurons, namely their projection onto the somatostatin interneurons, is the one that plays the most important role in spatial learning of location of reward. So in summary, I just want to close with a positive note, let's say, to what can biophysical models or simulation help us with. So I think that the work of my lab has contributed, and others, has contributed largely into generating a large variety of computational models, most of which are publicly available and can be used by the community. We've generated a large number of predictions that have steered the interest of the experimental community, and many of them are already verified experimentally, leading to new discoveries. And I'd like to think of what we do as a bridge between the experimental world and the theoretical work, a bridge that is much needed, and one that can lead to a lot of interesting predictions, explanations, and new experiments. So with that, I would like to thank the people that did all the work. Alexandra is the person that did the first project that I presented, and Spiros is the person that did the second project. Our funders and our collaborators, of course, and you for your attention. Thank you.
Great. We have lots of time for questions. I encourage especially trainees and postdocs to come up and ask.
Hi, thank you very much for the great talk. I have maybe a little bit of a naive question. So you were talking about challenging the point neuron dogma. And my question would be this two-layered artificial neural network, wouldn't that be by default be better than a single-layer neural network? What is the criterion where you say this is a sufficient fit, this is a good fit?
That's a very good question. So I don't think, of course, as you increase the complexity of the network, you are expecting to get better performance. So my claim here is not that we try to find a better classifier, let's say, right? Because if I use an SVM, it might be even better. If I use a hidden Markov chain, it might be even better. So the goal here is not to find a better fit. The goal here is to come up with an abstraction that incorporates what we see in these interneurons. And we do see nonlinearities in their dendrites, both positive and negative. So I would not phrase it as a matter of a better fit. I would phrase it as a matter of a better description, a reduced one, a simple one, but a better description of the reality.
Great, thank you very much.
Hi, Yiota. Hello.
Hi.
Thanks. I'm wondering, to reproduce the VIP-mediated plasticity work, the last story, what kind of learning rules do you need? Is a Hebbian learning rule enough, or do you need a coordination of homeostasis and maybe consolidation?
Okay, so that's a good point. So the model with the plasticity rule was used in the first work, not in the second work. In the second work, essentially what we implemented is just an increase of the weights between the CA3 input and the pyramidal cell. So it did not have explicit plasticity rules. All we did was to assume that if a cell happened to be firing within the location of the reward but did not produce a strong enough output, then you clump up the weights. So you increase those weights and you have new place cells joining the representation of the reward location. It's very much like what Jeff McGee presented with this 3-second rule, that you have new place cells joining the representation that were silent before. So I can't tell you about the specific plasticity rules. We have to put them in there yet first.
Thanks for a great talk. I was just wondering, so one other thing that we know about those different types of interneurons is their relationship to different aspects of the local field potential control. And I was wondering if you could comment on the extent to which your models can make predictions about the role of those different cell types in controlling local field potentials and linking those to behavior.
Right, that's a great question. These models do not incorporate the local field potential. There are ways to do it if you use neuron in biophysical simulations. They're not extremely straightforward, right? But I wouldn't be able to comment on that because I haven't actually done it. So other people may have, sorry.
Is it? Yeah. I was really interested in your two-stage neuron model, and I was wondering, because a lot of people use like normal point neuron models, especially integrated fire neurons, because they can get analytical solutions.
Right.
So has anyone tried to make analytical solutions or analytical calculations for the two-stage neuron?
Not to my knowledge. I don't think so. No. Although for such a model, which it's a simple one, right? So you have one type and two types and just a few neurons, maybe it would be possible, but I'm not aware of anyone that has actually done that. Maybe you should do it. That'd be great. Which one?
Okay, so yeah, I was wondering on what time scale the active properties of the dendrite would change. So you showed superlinear summation and sublinear summation, and that could, for example, be used to code additional. Well, okay, so it could have a computational purpose,
Absolutely.
but then it would be important to know if it changes on the same time scale as synapses, for instance. So are there any insights?
So you mean if the two types of nonlinearities, they operate on different time scales?
For instance, or if one cell type would sublinear
Right.
to sublinear.
Okay, yes. So in the experiments that I showed, the time scale is the same. So the synapses come in the same firing rate, so they're identical. So in the pyramidal neurons, there was evidence that sublinear dendrites can become supralinear. This is the work of Attila Lozonsky again back in 2008, I think. And it was done through the regulation of the A-type channel by decreasing the A-type conductance. So it is highly likely that in such a case, we could also regulate these nonlinearities and switch the sign of the dendrite. It is possible. I don't know if it happens and under what conditions, but it is possible.
Oh, that's great.
Yeah. Yes.
Great talk, thank you. I have two questions, but the first one is on the first part and whether you could comment on the role of spines or the absence of spines in the integration in fast spiking neurons. And the second question is on the second part of your talk, whether your model could make a prediction that inhibiting somatostatin will be an equivalent to activating VIP in 10 neurons in the rewiring of the place cells network.
Great point. So spines. We did not incorporate spines in this model, but definitely what the effect, the difference would be, let's say, compared to pyramidal neurons, is the compartmentalization. So you don't have as much compartmentalization of calcium signals, let's say, that you have in the dendritic spines in pyramidal neurons. You don't have that in the interneurons. But then on the other hand, you don't have an MDA conductances in these particular types of interneurons, right? It's because others do. That would be an important loss, let's say, with respect to compartmentalization. So in both of our models, the C1 pyramidal and this one, there were no spines. So the effect on the nonlinearity is not because of the spine, it's because of the channels of the synaptic conductances. So the second one as to whether inhibiting some would be exactly the same. We've done that, I believe we've done that in the model, and it was not exactly the same because the VIP don't only project to the some, they also project to the basket cells. So you have a biregulation of multiple inhibitory neurons. Definitely the somes also contribute to controlling the coupling between the cell body and the apical trunk. But for this particular output, they were not the same. It was not the same end result.
Thank you.
It's in the paper, you can check it out.
Hi, Yiota.
Hi, Balázs.
So I have a question on VIP, a little bit similar to Quentin's. And I was wondering what more details the model can tell on the circuit. For instance, there are positively and negatively modulated VIPs. How do they relate to CR and CCK? Are they both? What happens if you inactivate just the positive, just the negative?
Right. So it's all in the paper, but there is no clear distinction between the velocity modulation and the protein that describes the cells. So positively modulated are mostly coloredenine, but you also have the CCK in that category to a smaller percentage. And the other way is true for the negative velocity modulated. They are both mostly CCK, but you may have some coloredenine in it. So it's not clear, and until we get some better markers, I don't know if we can discriminate between the two. But we did simulate, so we simulate these percentages, right? And we did simulate the effect of cutting one versus the other. It's all there. The main result is that the CR, which is the main disinhibitory contribute, is important for spatial learning, if I can summarize it.
Thank you.
We have time for one more question.
Hi, thank you. Can you comment on the fact that there are many dendritic branches? So I guess my question is, one is, when we talk about dendritic spikes, do they propagate throughout all the branches? What does your model say about it?
In interneurons, you mean? In the interneuron model?
In either, I guess, yeah.
So there is definitely less compartmentalization in the interneuron than the pyramidal cell. So there is crosstalk between multiple branches. We haven't actually characterized this extensively, so I can't really say that it goes into all branches. My guess would be no, because you have a very distal one and very thin ones, but there is definitely more crosstalk than there is in pyramidal neurons.
Great. Let's thank the speaker for a wonderful talk.